German naturalist Fritz Müller hypothesized in the late 19th century that two or more species might converge in their colour patterns if these colours were to serve a particular purpose. For example, if one species had a preferable trait such as bright coloration to warn off predators, then this coloration might be taken on by other species in order to limit predation of their species. A recent report written in 2011 by Marcos Alexandro and his colleagues questions why two differing fish species share the same colour patterns within a shared environment. Alexandro and his colleagues attempted to find out if food competition drives catfish mimicry by studying the community structure in a diverse group of armoured catfish that inhabit the waters of the Amazon River basin. Shown here is a prominently marked catfish, Corydoras howard-schultzi, one of the mimetic Amazonian species studied by Alexandro and colleagues. The authors wanted to know whether the identically shared colour patterns among coexisting species were resulting from shared ancestry or from convergence due to mimicry. In order to determine this, the authors developed a phylogenetic tree of 425 species and compared the relatedness of specific species that occurred together in mimicry rings. Furthermore, the researchers wanted to know whether the fish differed in diet or whether this had any effect on the species coloration. They estimated fish diet using two metrics: snout shape, indicating how they acquire their food, and also stable isotope ratios of nitrogen and carbon in their body tissues. The researchers identified 52 species belonging to 24 different mimicry rings, composed of 2 or 3 species each. They also stated that a comparison of geographic location showed that 92% of mimetic communities are composed of evolutionary distinct species. This proved that most of the species studied were phylogenetically distant. Shown here is the relationship between fish species in the Amazon basin, including co-mimics. The figure shows each lineage and percentage of mimetic species belonging to each. The researchers then clarified that fish coloration similarities were not only determined by influences of genetics, but also by their diet. They found that the species within mimicry rings diverged considerably in their diets and morphological traits, suggesting that competition for limiting resources also affected the coexistence of the species. The results stated that the divergence of isotopic signatures reflects the dietary segregation between long-snouted compared to short-snouted species. This is represented in the table of isotope statistics here. There were no updated versions of this paper. However, in line with the work of Alexandro and his colleagues, a study conducted by David Penning and David Kikuchi on the topic of competition and the evolution of imperfect mimicry provided a different viewpoint on the concept, recognizing that mimicry does exist and is a strong example of evolutionary adaptation, but is often imperfect, and they stated that competition within species was the reason for this.